Okay, good morning, everyone. Quick reminder, this week you have your second homework assignment that will be due this Friday night. I will post it later today, and so you can look for that. You also have your weekly quiz that will be due on Wednesday morning. Okay. Just to get everybody sort of used to that routine, right? Pretty much every week, we have a weekly quiz due Wednesday morning at 10:30. So look out for that. And this week you have your next homework assignment due by Friday. Right? I was very pleased with all of your hard work in your first article summary that you submitted last time. I really enjoyed our first article share day, and hopefully all of you enjoyed that experience as well. I look forward to looking up some of the papers that you identified, because I think many of them were very interesting topics that I want to read a bit more about. So I admit part of the article share is so that you guys go out and find interesting papers, right? And then when you show them to me, oftentimes I'm like, wow, that's great. I want to read that and learn something more, myself, right? Okay, let's continue with our lectures. So last time we were looking at how viruses or certain types of viruses are responsible or have a causal link with different types of cancer. And we said that around 20% of all cancer types, there is strong evidence that viral infection of a particular type of virus can be a strong influence on the development of that cancer. So today, let's take a look at what is really happening then when these viruses infect the cells of the human body. What kinds of changes take place that actually start one of your normal human cells down the pathway of transforming. So let's review some key concepts since it's been a few days since we had a lecture. We said that some cancers are caused by specific viruses, not all of them. Right? So for example, some standouts are things like cervical cancers are certainly indisputably linked with viruses like HPV, whereas other types of cancer, like lung cancer, right? There's very little evidence of viruses causing lung cancer. The real standout risk factor is smoking. Okay, virus particles, again, as a review, are not really living things. They are non-living biological material. When they infect cells, they're designed to hijack the host cell and cause the host cell to make many more copies of the virus. But on occasion, that process of infection encourages the host cell to transform, begin to take on different types of transformed phenotypes. So transformed cells lose control of critical cell functions. For example, control over proliferation, control over maintaining proper morphology or the differentiation state, and things like losing dependence on being anchored to other cells in the tissue. Right? So transformed cells can eventually function in an anchorage independent kind of manner. We also learned that continued expression of the viral genome in an infected cell is required to maintain the transformed cell's phenotype. In other words, if the virally infected cell is starting down the path of transformation, you can reverse that at an early stage if you can somehow remove the viral components. And then we saw in the sort of very famous experiment done by UC Berkeley researchers that if you reverse the process of the viral influence within maybe the first few days, you can actually see those transforming cells revert back to the normal phenotype. But again, that only works up until a certain point, because if the transformed cell becomes too messed up, then there's no turning back. And transformed cells then, if they continue down this path, they become tumorigenic. So let's talk about mutations, because I think mutations are, of course, at the heart of what cancer is about, right? So these viral infections that are somehow associated with cells transforming, it must have to do somehow with mutations. If we look at this, this is a nice cartoon because I think it nicely illustrates the difference between what we call germline mutations and somatic mutations. Somatic mutations are the ones we typically think of as the ones that are brought on by exposure to something like a carcinogen in the environment, or brought on by exposure to something like the unfortunate infection by a particular virus type, infecting a particular type of cell in your body. Importantly, germline mutations, right, are different. Why? If you look at this here, let's say this kind of represents 3 generations in a family. Before the start of the first generation, because let's say the first generation are the parents in this square here, and then the children of those parents are in the second square here, right? That's why it's labeled parent and offspring. So if you go 1 generation prior to that, you have the grandparents, and the grandparents are each contributing, right, gametes. So, from grandpa, this would be sperm. From grandma, right, this would be the oocyte or the egg. And together, they form a zygote. Right? So it's kind of nice, even though this is oversimplifying it. Obviously, it's sort of representing the fact that you have 2 genomes or 2 haploid genomes in gametes coming together to form a diploid zygote, right? And this is the new individual here. During that individual's life, right, these stem cells that originated from the original fertilized egg, and of course through many, many, many rounds of division, have now populated and become all the cells in the body. Let's just say, for example, you follow one of these stem cells, right? And one of these stem cells, through many, many rounds of replication, right, formed, let's say, this particular multipotent stem cell. Maybe this is, for example, the type of stem cell you'd find in the bone marrow. That's then going to further differentiate and make the different types of cells that you see in your blood, right? So let's say the light blue one, let's say it divides and everything's fine, right? So it keeps dividing and you have these different intermediate cells, right, that then eventually create the fully differentiated cells. And these are the healthy versions of them. They have the normal intended genome in them. But if, let's say, in the process of dividing, you get a mutation, so we'll call this mutation B, right? And it happens at the level of this particular multipotent stem cell in the bone marrow, right? So when it divides, it has a daughter cell that carries this mutation in it. Maybe that mutation was a result of some carcinogen. Maybe this mutation is just a result of an error in DNA replication. And so the point is, that particular intermediate stem cell then, as it further divides and populates your tissue, like your bone marrow, with fully differentiated cells, you will have a subpopulation that are all carrying this mutation. Right? Hopefully that mutation is not a big deal, but this is a cancer class. So what we're saying here is potentially, right, what you're seeing is a population of cells that might be more predisposed now to transforming. The important thing, though, is because this just happened in a somatic cell, in the body somewhere, it does not influence the germline. Right? So you have in this case, the stem cells that originally made the body, some of them give rise to your germline cells. And these germline cells then, what if they ended up having a mutation? So mutation A happens in the germline cell production, right? Then that means the version of the gametes that are created by this germline cell, they carry the mutation. And that means now, right, when the parent then mates and gives rise to offspring, right? So this is the gamete from the mate here, right? The zygote that forms carries the mutation now, right? So in many cases, when we talk about sporadic cancers, sporadic means sort of out of nowhere, there was no history of it in the family. We are talking about mutations that happen somewhere in the body, like mutation B. So it only affects that particular individual, whereas the children that they have would not be affected. But there are certain mutations that happen in the germline, and those are passed on. So those would be hereditary cancers, right? In other words, the children, the offspring here, every single cell in their body has the mutation to start with. And so if this is a mutation that somehow predisposes them to developing further mutations, further transformations, right, that means the offspring are more susceptible to cancer, et cetera, and et cetera. Okay. So when we are talking about viral infections, then, I think you can imagine that by far the majority of the time, we're talking about viral infections of somatic cells in our body, right? For example, the epithelial cells that line our respiratory tract or our GI tract, right? So the fact that those may mutate and become precancerous lesions has no impact on what we pass on to our kids, right? Because our germline stem cells, in this particular case, were not affected. Right? So what is present in some viral genomes that cause the transformation of host cells and ultimately the formation of tumors? Right? I showed you this picture last time when we introduced the idea of viruses just being these non-living packages of biological material. We said there's some genetic material, there's the presence of some proteins here that make use of this genetic material in terms of hijacking the host cell, and then it's packaged up in an envelope that has different types of proteins like these green spikes that are used for the virus to stick to and fuse with target cells. But we said the important thing here is the genome itself of the virus is small, right? In a typical papovavirus, it's only about maybe like 5 kilobases, 5,000 base pairs. And I said last time, it's really intriguing to think that when you look at this image here, right, and you know that this is a chain of A's and T's, C's and G's going around, and there's 5,000 of such base pairs in this chain, that in this, right, are different sequences that code for all the components necessary of the virus particle, and something in there that can transform a healthy cell in your body to make it become tumorigenic. Right? So the earliest historic examples of these came from researchers who were looking at virally infected cells, and the obvious thing was to look for the presence of proteins that don't belong in a normal healthy cell. So SV40, again, is a simian virus number 40. And we said that SV40 causes monkey cells to transform, all right? Most of the time, this SV40 virus, right, succeeds in hijacking a monkey cell, and then the monkey cell just becomes this factory that makes more virus particles, leading to the destruction of that particular cell. And if that happens, then you're not going to get cancer, right? Because that cell ceases to exist. It dies. But every so often, you will have examples of a monkey cell that's infected by the SV40. And for some reason, the virus particle doesn't carry out the program successfully, and it ends up just kind of transforming the cell. Okay? So early researchers looked at cells that were infected by SV40. And in particular, what researchers did was they would infect non-monkey cells with the SV40 virus, because in non-monkey cells, right, the SV40 virus does not succeed in hijacking and replicating itself because it's designed, right, to do so in monkey cells. But in other mammal cells, that replication is kind of handicapped, right? Because the proteins inside of, let's say, a mouse cell or a human cell, yeah, they're all mammal proteins, right? So we're all kind of related, but they're different enough that the SV40 virus can't carry out its full replication potential. So in those cells then, right, SV40 virus causes transformation. And what researchers saw when they stained, for example, mouse cells or human cells, they could identify that there was the presence of a protein in these cells that didn't belong, right? And so since they figured that this protein must have something to do with the formation of tumors, responsible for transforming these cells here, they called them T antigens, right? So if you had certain antibodies that you're using to kind of probe different cells, and in this case, those antibodies are linked to maybe fluorescent molecules so you can make them glow, or they are linked to other molecules that you can use to chemically stain. In this case, you could see the presence in these human cells of this mysterious antigen. And they realized that this was some sort of protein that was made in these cells, responsible somehow for transforming. So they called it SV40 large T antigen. All right. So here's a thought question then. We said that if a virally transformed host cell continues to express certain viral proteins like large T antigen, long after the viral infection has been, let's say, beaten, right? Because at a certain point, the SV40 virus infection might be successfully defeated by the immune system. And yet these cells that started transforming, right, if the process has gone on far enough, right, they continue to transform and they continue to express large T antigen. Right? So if that's true, then what does this suggest has happened to the viral DNA? Because we no longer have virus particles around, right? And yet these cells are still making large T antigen. What does that suggest? It suggests that the viral genome has somehow integrated itself into the host genome, so that even though you no longer have infectious virus particles being made, right, and the immune system has succeeded in beating the infection, the viral genome is still there because it's integrated into the infected host cells. So DNA viruses and RNA viruses have different integration mechanisms. So let's take a look. We'll do some sort of comparing and contrasting here. DNA viruses integrate randomly as fragments. So here you have, as an example, HPV, right, human papillomavirus that gives rise to cervical cancer. So if we picture that the genome of HPV, right, are these small little compact genomes here, we can already intuitively imagine that the genes contained within the HPV genome, even though it's a small little genome, only a few thousand base pairs, there must be certain genes that are more important for actually the transformation process, right? And certain genes that are more responsible for just doing things like making the host cell make more virus particles. So we call these early regions of the genome. So the dark red means certain genes that are more responsible for encouraging transformation of the host cell, and certain regions of the viral genome that are what we call the late region. Now, you might already ask, why would there even be genes that are early region genes that transform the cell or encourage transformation? The key here is, remember, a typical cell in your body, like an epithelial cell in your respiratory tract, or an epithelial cell on the lining of the reproductive tract, like the cervix, right, normally would not care about going through the cell cycle heading towards mitosis, right? A fully differentiated cell is very happy just staying, like in G1, for example, just doing its normal epithelial cell functions. Right? So if a virus came along and got into that cell, right, and the virus wants that cell to actually be actively engaged in doing the types of activities that involve, for example, the production of more DNA, or the production of proteins that are essential for things like increased protein synthesis. Then you can imagine that part of doing this is actually convincing the host cell to no longer be happy just in G1, that the virus, as part of hijacking it, wants to shift the fully differentiated cell into S phase, right? Because in S phase is when DNA replication happens, and you have then the synthesis of many important proteins that are involved in what the virus would need in order to get the cell to really start becoming a factory, making more virus particles. So that's why, if you're talking about shifting a cell that was happy in G1, now you're encouraging it to start moving through the cell cycle, right? Eventually, if a cell that's going through the cell cycle will end up in mitosis, right, and proliferate. Therefore, we already can see that part of the virus's strategy of getting the cell to do what it wants it to do is to kickstart the cell through the proliferation process. But the key is, normally, right, the virus wants the cell to move into a phase where it helps the virus produce tons and tons of virus particles. The host cell never makes it to mitosis, right? Because it's then stuck in this hijacked phase where it's making tons and tons of virus. Eventually, the cell just dies, right? Because all of its resources are used up making the virus particle. So there are certain genes then in the virus genome that push the cell cycle forward. That's the early region genes. And then you have the other genes in the virus genome that once the host cell is in that very desired stage that the virus wants it to be in, then the late region genes kick in, and then that makes the host cell make a ton more virus particles, right? So why is that important? Because if you think about, again, the genome of the virus particle here, right? DNA viruses have very unstable genomes because they're just little rings of DNA, right? So what can happen is that these little rings of DNA can break, and then they can join up again, and you can actually have these much larger rings that represent multiple copies of the genome. In other words, concatemers, right? Just because the DNA is falling apart, and then you have the sticky ends of the DNA. If another ring of DNA fell apart, then maybe those sticky ends would come together, and you'd have this large concatemer. And of course, this concatemer itself breaks and linearizes all the time. So you get this messy hodgepodge of different versions of the viral genome now inside the host cell. Okay. And these different regions then, these different random fragments, will recombine with host chromosomes. So this gray stretch of DNA is, let's say, a stretch of a chromosome inside of a normal, healthy human epithelial cell. The random fragments of the virus particle will then integrate into it. Right? So in many cases, because remember, one host cell is not being just infected by one virus particle, right? There are potentially hundreds, if not thousands, of viral particles, all dumping their DNA into the infected cell. So you would have a massive mess of different fragments of viral genome integrating all over the place inside of the host cell. In some cases, you would have intact genomes of the virus. In other words, all the information that the virus needs to carry out the early and late stage hijacking of the host cell is present, right? And those are the ones that will then really hijack the cell, right? But scattered throughout, you would have these random fragments that might only have certain parts of the transforming genes and not the late genes, right? What would happen then if in reality, the ability of the viral genome to successfully hijack the cell is compromised because maybe the fully intact versions of the genome, maybe they don't integrate successfully, or they integrate in places of the host genome where they don't really work very well, they're not transcribed very well. But let's say you have these smaller pieces of the early region part of the genome that by chance integrated in key parts of the host cell where they are actively transcribed. Therefore, you get the effect of the host cell being shoved through the cell cycle, right? But because the viral genome is not successfully hijacking it to make more particles, what happens? It's now moving through the cell cycle, right, and proliferating. And that can then contribute to transformation, because in a ton of other places throughout the chromosome, you also have the normal chromosomes being disrupted by the viral genome. So in HPV, what they found is that over 99.7% of cervical cancer tumors carry fragments of HPV in the tumor cells. And so that's exactly what's going on here. It's just the unfortunate situation where HPV infection in some cells of the epithelial lining of the cervix, right, got a bad combination where early region genes were integrated in places in the genome where they're actively expressed, but those fragments did not have the rest of the viral genome, so that you don't actually get the production of viral particles. And in some crazy sense, it would have been better if the intact virus genome was integrated, because in those cells then they would have just died and given rise to virus particles. It would never have led to transformation and cancer. Okay. And not surprisingly, when pathologists, oncologists study what fragments of the HPV virus, right, tend to be the ones that are found in almost all cervical cancer tumors, what do they find? It's those early region fragments, right, that are present, right? Because that's what eventually led to those cells transforming and becoming cancer. Okay, so we can consider these regions, the early transforming genes then, right, to be oncogenic. They cause cancer. And so these fragments we can think of as viral oncogenes, right? In other words, some viral gene that causes cancer when it's part of our cells. So that was DNA viruses like HPV. The other main group of viruses, of course, are the RNA viruses. So kind of canonically or classically, we think of RNA viruses as being reverse transcriptase viruses, because their information is encoded as RNA that's being carried around by the virus particle. Once this virus particle infects the cell here, so this is now your cell membrane, and this space over here is the cytoplasm of your cell, and then this tan color membrane is the nuclear envelope, so this is now inside the nucleus on the right here. When the RNA is released into the cytoplasm of a host cell, it has to be reverse transcribed, right? So the RNA contains other proteins in it, such as reverse transcriptase and integrase, that takes care of making a DNA copy from the RNA. Again, that's why it's reverse transcribed, because normally we think of the direction of information flow as starting from DNA, and then you make RNA, and then it leads to something like a protein, right? But in this case, the viruses have reverse transcriptase enzyme that can make a DNA copy, and you end up with a double-stranded DNA copy, a perfect copy of the intact virus genome, but now in double-stranded DNA form. And that double-stranded DNA intact viral genome then integrates into our genome somewhere randomly, and we call this a provirus, right? Because now, depending on where this is integrated, if this DNA stretch is transcribed and expressed, you will make new viral RNAs from it, and then those viral RNAs will be processed into mRNAs that then get translated by the cell's ribosomes into all the different viral proteins that then get packaged up as new virus particles. Right? There are some RNA viruses that are even more clever, because you might say, wait, if it's RNA to start with, right, why does it even have to get turned into DNA and put into the genome? Because why can't our cells just be hijacked and make proteins, viral proteins, directly from the RNA? Right? In some viruses, that's what happens. You might be really interested to know that coronaviruses, like SARS-CoV-2, is an example of an RNA virus where its RNA skips the whole nucleus step of infecting the host cell, and the host cell is just forced to make protein products directly from the RNA to make more viral particles that way. Okay, so it may very well be that even though SARS-CoV-2 is a terrible, terrible virus, right, it may be that eventually, over time, we never discover that there's any link of SARS-CoV-2 with cancers because it skips this whole genomic integration step. Okay. So let's go back to our story then about Peyton Rous and his Rous sarcoma virus, and take what we've just been talking about the last couple of slides and kind of follow the story of what happened next. So remember, the Rous sarcoma virus is a tumorigenic virus, right? This was the virus that was isolated from chicken sarcoma tumors that Peyton Rous had access to. Right? It is closely related to other types of avian or bird viruses, for example, avian leukosis virus, which affects the white blood cells, leukosis, but it is non-tumorigenic. In other words, birds who are infected by ALV, they get sick, right? They might die, but it's not because of developing cancer. Right? If you compare the genome of ALV, right, with the Rous sarcoma virus, the RSV, and you line them up. Now they're both about the same size, relatively speaking, right? And you would not be surprised to find that certain parts of the genetic sequence line up very well. The genes that make the core proteins of the virus particle, the genes that code for things like reverse transcriptase and integrase, right? Those are almost identical between the two. The genes that encode for the envelope proteins, the outer part of the virus with all the glycoprotein spikes, those are nearly identical as well. But what early researchers saw was, wait a minute, the Rous sarcoma virus has this extra brown genetic sequence in it here. Well, I mean, obviously it's not brown, but it's this extra sequence here that in this cartoon is represented in brown. So the researcher said, aha, it must be this extra piece of genetic information that is what gives the Rous sarcoma virus its transforming capability. Right? So this is some kind of oncogene, again, a viral oncogene, and they called it SARC, right? Because it causes sarcomas in the chickens. So RSV has what we call this viral SARC, right? So viral gene that causes sarcomas. And so the question is, how did this VSARC gene arise? How did the Rous sarcoma virus get this, right? Or for that matter, how did any part of the viral genome originate? Where did it come from? I already gave you this sort of analogy of the buffet and the sandwich. You know, I love this analogy. These food analogies work really well for me, right? But remember, we said this idea is that the sandwich is a subset of ingredients from the buffet that are prepared slightly differently so that they work now as a sandwich. So the bread is sliced into slices instead of broken into chunks. Likewise, the cheese is in slices. Likewise, the lettuce is torn into little pieces, right? Things like that. And we said that that came about through a coincidental combination of different components from the normal cell, the buffet, being altered at the right time so that they could come together and make something that stood by itself. And importantly, we said the virus is still, I'm not sorry, the sandwich is still dependent upon the buffet existing, right? If the buffet goes out of business, right, then there's no more ingredients for the sandwich. Right? But of course, the point here is that the little bits and pieces that gave rise to viruses billions of years ago were not the actual protein molecules itself, right? In the example of the sandwich coming from the buffet, right, we're talking about the bread being shaped differently or the cheese being shaped differently. But really, right, in order for the virus to continue, right, or you can think about it, in order for other cooks in the kitchen of the fancy Waikiki buffet, you know, restaurant here to be able to make sandwiches, it's the knowledge of how to make these mistakes. In other words, these mistakes cease being mistakes and they actually become the intentional information on what you do to the bread or what you do to the cheese to make the sandwich. So if we said that billions of years ago, parts of normal cells, right, that's the buffet, gave rise to viruses because there were little scraps that became these viruses. What are these scraps? The scraps, of course, are fragments of the genome, fragments of the original genome of the host cell, pieces of RNA and DNA that somehow got altered and became part of the virus's genome. So the Rouse sarcoma virus, we said, has this V-sarc, this viral sarcoma-causing oncogene in it, right? And so researchers, once they realized this, this is back in the 1960s now, they started looking at different examples of RSV infected cells, right, to see if they could find evidence of this protein, whatever this protein product is, this sarc, right, that's being made. So they designed things like antibodies against it to be able to probe cancer cells. As part of testing these antibodies out, of course, you need to have a control, right, to make sure that whatever staining from these antibodies you see is actually meaningful. So they would go ahead and test it on something like a negative control, like a healthy cell, right, like a healthy chicken cell, right? And they were shocked because what did they find? They found that the probes that they used to find this V-sarc in these cells was also present in the uninfected chicken cells as well. And they could find it in mammals as well. That means this sarc gene is already present as part of our normal genomes, right? And you say, what? Why would we have a tumor-causing gene that throughout evolution is actually kept in our genome? Right? And hopefully you realize it's because, whatever function that the normal sarc has in our genome is important, and it plays a role in the normal cell cycle, pushing the cell cycle forward when it should be pushed forward. But if you have a version of this sarc that gets altered in a way, right, no longer behaves the same way, that might contribute to out of control cell cycling. So the normal sarc that's found in our genome is called C-sarc, or in other words, normal cellular sarc. Okay. So what are the implications of this? So the implications then are that throughout evolution, right, mistakes in the form of normal important genes in our bodies being altered in a way and kind of shed in a way as little scraps, right? That is what gave rise to viruses. And just like the sandwich had to only happen with the right combination of mistakes of bread and lettuce and cheese and whatever, right? Viruses only evolved and came about because of the right combination of mistakes, right? And that right combination now allows them to exist as their separate kind of entity, not alive, right, but capable of wreaking havoc on our cells. I often think about viruses as being this cloud, right, that has followed us through evolution. All species, complex species of animals, right, have this cloud of viruses around them because they represent all these little mistakes over evolution that have resulted in these self-sustaining little things that can actually depend upon infecting our cells to keep them going, right? It's kind of like in the old Charlie Brown Peanuts cartoons. There's that one kid, Pigpen, right? And Pigpen's always dirty. He's the one who always looks kind of dirty. But importantly, wherever Pigpen goes, there's like this cloud of dust that follows him around, right? Pigpen is like the evolutionary history of us. Right? And if Pigpen were suddenly to disappear, right, that cloud of dust in the cartoon would probably just dissipate and disappear as well. And sure enough, the viruses that plague our human population, they count on us still being around in order to still exist as this cloud. Right? If people suddenly disappeared overnight, right, so would things like SARS-CoV-2, so would things like HIV. Okay. So let's look at how these virus particles may have acquired oncogenes. Let's say a long time ago, looking at the example of birds and chickens again, there was this avian leukosis virus particle, right? Again, this is a virus particle that makes chickens sick or birds sick, but it does not cause cancer. Let's say one day, particular virus particles infected a bird cell. So they made a double-stranded DNA form, a provirus, and then that provirus, will randomly integrate inside the host cell's genome. Let us say this region of the chromosome that we're zoomed in on up here happens to be the region of the genome on a particular chromosome where the C-Sark gene is located. Right? And by awful chance, the provirus randomly integrated right in front of the C-Sark. What would happen then is as the provirus gets expressed and replicated, you can imagine, right, that the replication process may also carry with it part of any downstream DNA in here. So you get different fragment sizes of the DNA that's replicated. Some of them would contain this C-Sark. Then this ALV virus that's made here now has this extra bit of Sark on it. At first, it's just the normal Sark that's found in the chicken. It'd be the green version, the C-Sark, right? But of course, viruses mutate very rapidly, very easily. So you can imagine then that some of these virus particles would start getting random mutations in different parts of its genome, and some of them by chance would get mutations in the C-Sark part that they're carrying around that then result in the C-Sark's sequence being different enough that now when it's expressed inside of a host cell that's being infected, it makes a viral oncogene version of the Sark that no longer behaves properly. And so that then gave rise to our Rous sarcoma virus, right? So now when the RSV integrates inside of a chicken or a bird, right, and its proteins are expressed, this little what do you call it, hitchhiking gene, right, which is the C-Sark that got randomly mutated into V-Sark, is also expressed inside the host cell, and this throws the host cell's cell cycle out of whack. So such viruses that have acquired oncogenes in this way, right, are considered fast transforming. In other words, if the original normal wild type C-Sark is a protein that's important for regulating the cell cycle, then you can certainly understand that if the function of that Sark is compromised in such a way, for example, if it's no longer behaving the way it's supposed to be, then the effect can be the cell cycle begins to happen pretty rapidly, right? And so oncogenes like this that involve a particular gene that's critical for the cell cycle, the result is the infected cells transform pretty quickly. They're fast transforming. You can actually use the presence of these fast-transforming viral oncogenes to help identify, what particular normal genes in the host genome actually play a role in proliferation, right? Because if we say the brown V-Sark here, right, is once it's messed up and mutated, it drives the cell cycle forward. If you didn't know that there was such a thing as an original C-Sark in the genome somewhere, you could actually use the fact that you know the sequence of the V-Sark and ask the question, is this a sequence that we can identify in the host genome to start with? And by doing so, you might actually identify another normal gene that is important in cell cycle progression, for example. And so such normal genes, the versions of them that we carry around, right, the normal versions, are what we call proto-oncogenes. So the implication is these are the normal versions of the genes that are not tumorigenic, they're not oncogenic, until something goes wrong with them, and they can become full-blown oncogenes. For example, mutations. So this is an example of some acutely transforming retroviruses and oncogenes. So these are all fast transforming examples of viruses. You don't have to memorize this table, but I wanted to at least give you an example of how these viruses, like Rous sarcoma virus, led to the discovery of critical oncogenes and proto-oncogenes like SARC. Right? And now, of course, many years later, we have a good understanding of what the actual protein product is of the normal CSARC that is expressed in our cells. So you can see here, these are different things like these non-receptor TKs. We will study what TKs are later. If you look at these different oncogenes, some names may seem familiar, like MYC is a very important oncogene, right? And again, if you've been following along, our normal genome has MYC in it, the normal cellular, well-behaved version of the gene, right? And MYC is a transcription factor. Some of these are what we call RTKs, or receptor TKs. And we will look at those as a very important set of pathways that control transformation. And other examples here, like the RAS genes that come from mouse sarcomas, so HRAS and KRAS, right, that function as small G proteins. And we will look at later what the function of G proteins are. So in essence, what we're talking about is that because this all has to do with the random integration of viral genomes into the host genome, causing random disruptions in different places, right? Not surprisingly, if you were to look at tumors that represent the end product of random integrations that happen to be the right spot to cause the cell to transform and become a tumor, and you were to then say, let's say we take 100 tumors of a particular type that were caused by random integrations of a virus of a particular type, and let's look at where the virus genome integrated in these tumors. And let's say that here is the MYC gene as part of the normal genome here. It has 3 exons in it. These little blue triangles represent different examples of where the viral genome was seen to integrate in, let's say, 100 different tumors. And what you can really see there is there's a selection going on, right? So in those cells that were unfortunate enough that it had integration of the genome, the viral genome, in front of exon 2 of the MYC gene, these are the transforming cells that then went on to form the tumors, the 100 tumors or so that you took the sample of to see where the integration took place. Right? So in this case, what it's doing is the integration is actually causing maybe uncontrolled expression of part or the whole MYC gene. Right? So this can then lead to an example where the MYC gene is not necessarily mutated, but what has happened is in the host cell, the random integration of the virus is causing a lot more expression of this particular MYC protein, making it oncogenic. Okay. There are other cases as well. I know we're almost out of time, so I'll just finish up with this example here. If you think about a gene, a typical gene that has a coding region, the transcribe region, and then a promoter, right? And there may be distal regulatory elements like enhancers where different transcription factors bind, right? So typically different transcription factors bind to different sequences in the DNA that they recognize. So these transcription factors are like these orange, I don't know why they're drawn like Hello Kitties, right? But they're orange Hello Kitties. They pile up here and they cause different transcription factor proteins to build up. Then that whole enhancer region is brought to the promoter. Right? And that's what then forms the right shape to recruit RNA polymerase there. And then the RNA Pol2 expresses or transcribes the gene. So think about this. If you have polymorphisms or mutations in different spots in this DNA, you can have different results. For example, a mutation in the coding region of this gene could really impact the eventual amino acid sequence of the protein, and that might compromise the function of whatever protein's made here. What if you had mutations, though, or polymorphisms in one of these transcription factor binding sites? Then the protein that's made here might still be the totally normal protein, but you might see a dramatic increase or a dramatic decrease in the expression levels. And in the case of the MYC that we looked at, what if this whole region in front of the promoter actually got replaced by a viral promoter that came from the random integration of, let's say, a provirus in here, right? That would also lead to loss of regulation of whatever this protein is. What if this protein itself is the orange Hello Kitty gene, right? In other words, this is the gene that codes for the orange Hello Kitties. Right? Then you might have out of control production of that protein. And because it's an important transcription factor itself, there's a feedback, right? Because now you will suddenly lose control of many other genes in the genome that are dependent upon the orange Hello Kitties to regulate them. So in this particular case, the transformation process of the cell might be slower, right? Because we're not talking about a particular proto-oncogene that now is being overexpressed like MYC, resulting in oncogenic activity. We're resulting in something more subtle, right? Like the loss of regulation of the orange Hello Kitty protein, which really has nothing to do with cell proliferation, but it's an important transcription factor that does control many other genes. And so the combined effect of the loss of proper control over a range of other genes might eventually contribute to transformation. So we say they are slow transforming. Okay, and then this is just the last slide. So again, just like the slide that I showed you before, these are examples of many cellular genes that were determined to be important in cancer, identified by seeing where different viral proviruses integrated in the genome. So MYC we already looked at, right? So because researchers saw that in tumors, a lot of times you would see proviruses integrated right in front of the MYC gene. That's how they identified that MYC is very important for transformation. Okay, and these are just examples of other ones as well. P53 is one that we will talk about a lot. It's a very important transcription factor, and loss or disruption of P53 expression can have profound effects on transformation as well. Okay,